It's a big pleasure to be here and introducing Shumpei Maruyama, my most senior student in the lab, about not to be my student anymore, for his thesis defense talk, the public section of our talk. And we have all our committee members here. And as most of us know, we have a public part of the defense where Shumpei is going to give about a 45 to 50-minute presentation, followed by public people not from his committee able to ask him questions, comments, etc. And then we will recess to a different room and have a continue with you. So let me introduce Shumpei. Shumpei has a very wide-ranging thesis. That's what you're going to see. Very, very wide-ranging thesis with a focus on symbiotic algae present in cnidarians. And in our case, in the white lab, it's in symbiotic sea anemones. And Shumpei started off, as many students do, with jumping on board to an existing collaborative project. We'll talk about this part of the thesis on lectin-glycan interactions and the glycans that exist on algae. This is a long-term collaboration that I have with Sandra Luzbin at University of Florida. But right away, I could tell that Shumpei was creative. And so I would say that Shumpei is wildly creative. And this started to show up right away when he was proposing experiments in that glycan realm that I won't spoil the surprise, but ended up being very, very nice, very, very nice work. So he's definitely a tinkerer and a thinker and a reader. So these are combinations of things that have the consequences of them are this wide-ranging thesis, which the committee is probably like, what the heck, right? Okay. So then he convinced me that he could study afterwards, which he'll tell you all about. Afterwards are kind of a fancy proxy, similar to what antibodies can do in terms of finding specific moieties on things. Convinced me that this was something that he wanted to try. High risk. He'll tell you about that. So then that he was working on that, and then he started to fiddle around thinking about the role of expelled algae. So all hosts release these algae sort of homeostatically, right? I need to control my population of algae, so I'm going to spit them out regularly. Started to think about those algae, and he's kind of fiddling around. He had students who was working with fiddle, and then he's like, I think I want to work on this, right? So we started to talk about expelled algae as inocular for future infection events, and then lockdown happened. Right, so he's already been to Japan, worked on corals, had some problems with that he may or may not tell you about in terms of just getting into work on adult corals. And then lockdown happened and hit again, so he couldn't tinker anymore. So then he was thinking and reading. And so then he pitched me a project that we'll tell you about, a perspective that I think is honestly going to be a seminal paper in our, because it's shifting the landscape for how we think about the ability to study algae in the laboratory. And so he'll tell you all about that. But he wrote it during lockdown. He's like, well, I'm sitting here wasting my time. I think I'm going to write this paper. And I'm like, all right. And he wrote it, and then he submitted it. And then the reviewers came back and said, we love it. And I was like, this never happened. This will never happen to you again in your career, having no revisions, right? No revisions. So that was a miracle. It was awesome. So then, let's see, I don't want to forget. So then he came back, he continued to work on this process of expelled algae that I'll tell you all about. All of these things have been done, including undergraduates in his work, and at least one, maybe Alanis is on. Oh, she's on good. Alanis is on good. There are at least two students who he has mentored in the lab, but he's also wildly creative in his interest in pedagogy and in the use of implementing course-based undergraduate research experiences, partially because training in IB and all of the great stuff we do with our GTAs. And he, during lockdown, sort of piloted some work for the big introductory biology course that we run, and then also used students in the invertebrate lab as part of one of his thesis chapters at Bill. And so he's really, again, wildly creative about thinking about ways to help undergraduates learn research and the process of research. He has written and received a very prestigious NSF postdoctoral fellowship, continuing work in Aptasia, but also in corals with my colleague Phil Clevis at HHMI in Baltimore. He's going to have a great time there. Phil is a fantastic person, and Shumpei is going to bring a lot to that lab. So I'm really, really excited for that. And that also includes a cure. So with all of that, we are very happy in the White Lab for you, Shumpei, but we're also already sad that we're going to lose you. But you won't ever be far away. You'll stay on the Slack channel. So with that, I'm going to hand it over to you. I think we lost the battery.
I can hear you. Yeah. Hear the headphones? Yep.
Must be a room.
And the serial number, hear me? There are some batteries on the desk right there behind the dog can. In the top.
The doc can, the doc can. Here we go.
Oh. I can hear, tell us out there.
Party exam.
Okay, it's been.
Okay. So take it away.
Okay. Thank you, Virginia, for that kind introduction. So I'm excited to share with you today what I've been working on for the past 6 years of my life and my PhD. And so my thesis is titled "The Role of Algal Cellular Physiology on the Onset and Maintenance of a Model Cnidarian Algal Symbiosis." Oh, sorry. So first, I want to start pretty broad and talk about symbioses in general. So symbiosis is when two unlike organisms work together to make something that's greater than the sum of its parts. And symbiosis, so in the past, it was kind of like this rare thing, this peculiar thing. But more and more, we're finding out that symbiosis is more like a rule of life. Virtually every organism engages in symbiotic interactions. And these symbioses often form the foundation of the ecosystems in which they are found in. So for example, we have plant mycorrhizal symbioses. This is a symbiosis between a plant and a mycorrhizal fungi. And they've really allowed plants to take over the terrestrial world, and they're really important for the function of forests. We also have annelid bacterial symbiosis. This is a deep sea symbiosis between a deep sea worm and chemosynthetic bacteria. And this symbiosis has allowed these worms to thrive off of hydrothermal vents away from sunlight. So there's ecosystems in the deep sea thanks to this symbiosis. And we have human bacterial symbiosis that we can all relate to. This forms the human microbiome, and more we're finding out how important this microbiome is for human health as well. And so today I'm here to talk about coral algal symbiosis. And this symbiosis forms the foundation of coral reefs. And here's a particular coral reef off the coast of Okinawa. You can see they form these vast structures. They look incredibly beautiful. They house a lot of diversity of fish and invertebrates, so very important ecosystems. And so they're important because they're hotspots of biodiversity. They're known as the rainforests of the ocean. They're important for fisheries, for local populations that depend upon them for a major source of food. And they hold a lot of cultural importance and significance for people as well. People are finding medicines in coral reefs, unique compounds that can be used to fight cancers, diseases. They attract a lot of tourism, so they're economically important in that way as well. And they also provide coastal protection in the event of hurricanes and storm events as well. So these are the partners of symbiosis. So first we have the coral, which is the host, and they belong to the phylum Cnidaria. And cnidarians include other organisms like sea anemones and jellyfish that can also engage in this symbiosis as well. Here we have the dinoflagellate algae, and the algal partner belong to the family Symbiodiniaceae. And Symbiodiniaceae is a very diverse group of organisms that also have symbiosis with not just cnidarians, but also a lot of other kinds of organisms. So in this symbiosis, the host provides protection for the algal symbiont. They also provide a highlight environment and inorganic carbon, both to maximize photosynthesis for the algae. And the algae in turn provide their photosynthetic products, so things like glucose, lipids, sterols, and also oxygen to the host. And so in this symbiosis, the coral really relies on the algae for most of its energetic needs. And here we have Aptasia. Aptasia is the model system that we use for the study of cnidarian algal symbiosis. And I'll talk more about Aptasia in a little bit later, but they also belong to the phylum Cnidaria, so they're very closely related to the corals. And I want to zoom in on the tentacle of these cnidarians. And so this is what that looks like, the symbiosis looks like in a microanatomical level. So on the top here and the bottom, we have the epidermis. This is the outer tissue layer. Underneath that, we have the mesoglia, which is a collagenous layer. And then the algae are found within the gastrodermal layer. And so the algae are within these host cells. So it's an intracellular, very intimate symbiosis. And I can't talk about corals without talking about coral bleaching. So this is a coral reef that has undergone extensive bleaching, and they're completely white because they've lost their algal symbionts, and the symbionts are what give the coral its color. And so this can be very devastating for corals. And this is caused by heat-induced breakdown of the symbiosis, and corals without their algae will starve and eventually die if temperatures do not recover. And that's because the corals, again, rely on the algae for their energetic needs. And of course, this has been exacerbated by the climate crisis. So here we have a chart illustrating the heat content anomaly of the oceans over the decades. And since the 80s and 90s, the temperatures have been slowly ramping up, not slowly, pretty rapidly ramping up. And this has caused global bleaching events. And global bleaching events are when entire reefs around the world are bleaching basically all at once because the temperatures are just too hot that year. And so we saw our first global bleaching event in 1998, the first recorded bleaching event or global bleaching event. Next one we saw in 2002 and then in 2006. So every 4 years we saw global bleaching events. And then 10 years we had a gap, but then in 2016 and 2017 we had back-to-back bleaching events, the first of its kind. And then in 2022 we saw our most recent bleaching event. And so this is what that bleaching event looks like off the coast of Australia. So this is a Great Barrier Reef, which is the largest coral reef structure of its kind. It's a structure you can see from space, and it's incredibly important. And this diagram is a little bit confusing because this blue here is the land, not the ocean. The white is the ocean. And so you can see these are the corals here. And so in dark red and red, you can see severe and extreme bleaching. And so this event was incredibly devastating for the corals there. Virtually none of the corals were untouched by bleaching, which are shown in blue here. So to solve this crisis, mitigating climate change is critical, right? It's the major driving force of coral bleaching, but it is hard to rely on that as the only solution because of political.
Yeah, we were barking up the wrong.
Sorry, can I ask someone to? Oh, there we go. Thank you. Sorry about that. And so as scientists, there's now a race for the development of solutions to prolong the survival of corals into the future. Maybe we can give corals a helping hand to bring them into a future when we can eventually mitigate climate change. And so to do that, we need to understand cnidarian algal symbiosis. So I want to talk more about the specifics of symbiosis and more about the stages of the symbiotic relationship. So first, we're starting off with the onset of symbiosis, when the partners first meet together. So here we have the algal symbiont. The algal symbiont here is in its swimming form. So the picture you saw before, they were all spherical, they're in the coccoid form, and they don't swim, but they switch between these two life cycles. So when the algae are swimming, there's attraction involved. So the algae get attracted to the host. This can be from chemical cues or visual cues. Once the two partners meet, there's cellular recognition involved, which can lead to selection or rejection of the symbiont. And I should mention here that the algae don't have to be in the swimming form for this to occur. This is more what happens in nature, but in the lab, you can add whatever algae and the host can take them up. So chapter 1 of my work will be looking at the glycans, the role of glycans on the cell surface of the algae during the onset of symbiosis. And I'll talk more about glycans later. So when everything works well, the algae are taken up by the host, and the algae switches to this coccoid form. So when the algae are found within the host cell, they're always in the non-swimming coccoid form. And they reside within this symbiosome membrane compartment. So it's an organelle within the host cell that they reside in. And so now we're moving on to how the symbiosis is maintained. And so the host immunity is modulated by the symbiont. We're not sure how this occurs, but this is how it happens, or this is how the symbiosis is maintained. Because usually when you have a foreign cell inside of you, you want to destroy it or get rid of it. But in this case, it's being tolerated. So the symbiont is being tolerated by the host. The symbiosome becomes very acidified to a pH of around 4, so a very different environment from the outside ocean environment. And there's an active exchange of compounds between the host and the symbiont at this stage. Chapters 2 and 3 will be looking at the role of nutrition during the maintenance of symbiosis. In chapter 4, I'll be using aptamers to try to probe the cell surface of the algae during the maintenance of symbiosis. And again, I'll talk more about those specifics later on. So eventually the symbiosis ends. It doesn't have to, but it can. And so this is illustrating symbiont expulsion, where the algae are expelled by the host cell. And I should mention here that this isn't the only way that the symbiosis can end. It can happen from the host digesting the algal cell. It can happen from the host cell dying all together with the algal cell as well. But here in my thesis, I'll be talking about symbiont expulsion. So this can happen from homeostatic release, where if the host cell might have too many algal symbionts within it and it's trying to control how many of them there are, they might just release them as a homeostatic mechanism, so a non-stress event. This can happen from symbiont rejection, where if the symbiont's the wrong type of species of algae, they can be rejected. And of course, it can happen from a stress response like in coral bleaching. In chapters 2 and 3, again, we'll be looking at symbiont expulsion and more so at what happens to the algae after they're expelled by the host. So to study all of this, we use Aptasia in the lab, and Aptasia is a model system for Cnidarian algal symbiosis. So on the left here, we have Breviolum minudum. This is the algal species that I've been using throughout my work. Breviolum minudum is also found in some coral species, but otherwise they're pretty closely related to the algal symbionts found in corals. And here we have Aptasia. The scientific name for Aptasia is X-Aptasia diaphana. But throughout my thesis, I'll be referring to it by its common name, Aptasia. And Aptasia can be in its aposymbiotic form or its symbiotic form. Aposymbiotic means it's symbiont-free. And the symbiotic, of course, is symbiotic, has its algae, so it looks brown. And there's several advantages of using Aptasia over coral for the study of Cnidarian algal symbiosis. And one is that they're very easy to keep in aquariums, which makes it possible for labs around the world to use Aptasia to study Cnidarian algal symbiosis. You don't need to live near the tropic ocean to study them. They reproduce asexually very readily, which leads to the development of clonal lines. So there's been clonal lines that have been used over the decades in the research of Aptasia. So it makes the comparison of studies over the years very feasible. And Aptasia can be kept aposymbiotic. So unlike corals, which will die without their algal symbionts, Aptasia can live indefinitely without their algal symbionts, which makes it beneficial for experiments where you can study things like the onset of symbiosis. You can add algae back to the aposymbiotic anemones. You can also compare the biology of the two states to understand symbiosis more. The algal symbionts are culturable. This isn't true for all of the algal symbionts of corals, so this is really advantageous for Aptasia. And it's very well studied, which leads to the development of protocols that are available. There's a lot of molecular resources like genomes, transcriptomes, and proteomes. So this is a very good system for studying the symbiosis. Okay, so this brings me to my chapters. So chapter 1, I'll be looking at the onset of symbiosis. Chapters 2, 3, and 4, I'll be looking at the maintenance of symbiosis. And then chapters 2 and 3, again, I'll be looking at what happens to the algae after they are expelled. And then, so we'll be starting with chapter 1, which are looking at glycans and their role on the onset of symbiosis. So chapter 1 is titled Heat Stress of Algal Partner Hinders Colonization Success and Alters the Algal Cell Surface Glycome in a Nigerian Algal Symbiosis. And this was just published in Microbiology Spectrum last week, so hot off the press. So if you're curious, you can read that there. And this is a collaboration, like Virginia mentioned, between the Weiss lab and the Loskin lab and the McRef lab. So all the glycomic data that you're going to see is thanks to the expertise of these two labs. So what are glycans? So glycans are sugar chains that are attached to other molecules. So here we have a glycan sugar chain, and these individual shapes are individual sugars. So a chain of them make up the glycan. And this is attached to a protein in this case. So when it's attached to a protein, it's called a glycoprotein. Glycans can be attached to other molecules like RNA, lipids, but I'll be focusing on glycoproteins for my thesis. And these glycoproteins are embedded on the cell surface of this cell on this image here. And there's a lot of cellular functions of glycans. They allow for the proper folding of proteins. They're important for cell-to-cell communication. So a variety of different functions. But on the cell surface, they're important molecules for immunity and self-recognition because they function as ID tags for different species and organisms. And here's an example of how glycans play a role in immunity. So blood type is determined by glycans. And so here we have blood type A has a square glycan, blood type B has a circle glycan, AB has both, and O lacks any of those special ones. And so you might know if you get the wrong type of blood donated to you, it can kill you. And that's because, if, for example, if you group blood A and you get donated group B to you, your cells will recognize these glycans as foreign invaders. And so it'll start destroying it. And so this is an example of how glycans play a role in recognition and immunity. So what role do glycans play in symbiosis? So glycans are on the cell surface of the algal symbiont, and I've depicted them here as circle glycans and square glycans. And lectins are proteins that are found on the host cell. And these lectins are glycan binding proteins, and they do so specifically. So this circle lectin will recognize the circle glycan, the square lectin will recognize the square glycan. So when everything goes well, the algae is taken up by the host cell thanks to these glycan-lectin interactions. And there's evidence that these glycan-lectin interactions are important for the maintenance of symbiosis as well, because they're found recognizing each other in there. And so how do we know that glycans are important for symbiosis? So here we have the native species of algae, has all of its glycans, leads to successful uptake. But if you modify the glycans, you can hinder the uptake and the onset of symbiosis. So for example, you can enzymatically remove the glycans, have a reduced glycome, can hinder symbiosis. You can mimic that by doing lectin masking. So here we have an exogenous lectin masking square glycans here that can lead to hindered uptake. You can manipulate the synthesis of glycans with drugs. So in this case, we've increased the amount of circle glycans, decreased the amount of square glycans compared to the native species. That can lead to hindered uptake. And incompatible species of algae also have their unique glycomes, which gives us a hint to figuring out what glycans are important for recognition and figuring out which ones are beneficial, which ones are bad for symbiosis. So through all this evidence, we hypothesize that glycans are important for the onset of symbiosis. So this leads me to my questions. How does heat stress of the algae affect the onset of symbiosis? We know that heat stress is bad for coral bleaching, but what about during the beginning of symbiosis? And then we asked, how does heat stress affect the algal glycome itself as well? And we predicted that heat stress would change the algae to have a reduced glycome because it's just a stress event. And so we expected this is what would cause any changes during the onset of symbiosis. So first, we looked at the onset of symbiosis. To do this, we did some inoculation experiments. So here we have the algae for the control treatment. It's just ambient algae grown at ambient temperature. They were added to aposymbiotic anemones, and then we got symbiotic anemones and measured the symbiotic density. For the treatments, we heat stressed the algae at 32 degrees Celsius for 3 or 7 days. We added it back to anemones and then measured the symbiotic density. And I want to point out here that only the algae were heat stressed. and the anemones were always kept at ambient temperatures. So in this way, we were able to tease apart the effect of heat stress on just the algae. And then to measure symbiont density, we use fluorescence microscopy. So here we have a close-up of a tentacle. And these red dots you see are individual algal cells, so it makes it very convenient for quantifying this. And these images were taken 48 hours post-inoculation, so very early stages of symbiosis. And we used ImageJ to draw out the tentacle, and then we calculated the area of that tentacle and normalized the data to symbiont density to attain symbiont density. So here we have symbiont density on the y-axis, and we have the different treated algae on the x-axis. And you can see a very clear pattern that heat stress negatively affects the establishment of symbiosis. But we didn't want to call it there because heat stress also reduces algal motility. So there's a confounding variable here. And this is important because there's a mix of swimming algae and coccoid algae in the way we did the inoculations. And so the aptasia are just sitting there. And after a while, the algae will start to sink. So there's a difference in the number of algae that are available to the aptasia in the water column. So that might be causing the results here. So we repeated these experiments with some controls to try to eliminate symbiont motility. And so one way we did that was with hourly resuspensions, where I just came in every hour, mixed up the algae within that inoculation to try to artificially inflate the number of algae that were available to the aptasia, try to mask any differences in symbiont motility. We also did the inoculations in the dark. We did them in the dark period of the light cycle, so at night. And this could eliminate algal motility as a factor entirely because algae only swim during the day and they don't swim during the night. But we saw the same pattern. So with heat stress, it reduces symbiont establishment or symbiosis establishment. So something else is going on here. So going back to the questions, how does heat stress of the algae affect the onset of symbiosis? And very clearly, it hinders the establishment of symbiosis. And so then we turned to the algal glycome. Could it be changing like this? So we first did some lectin staining. So these lectins are fluorophore conjugated lectins, so they fluoresce under certain wavelengths of light, and these were provided by the Lozgen lab. And the two lectins we used were CVN, which is a high mannose-specific lectin, and SNA1, which is a sialic acid-specific lectin. And I'll talk about the significance of these a little bit later. But to do this, we incubated the lectins with the algae. And then because they're fluorescent, you can measure the fluorescence of the cell to determine how much of the lectin bound to these cells. And to do this, we did flow cytometry. Flow cytometry lets you put a sample of cells through a tube, and they pass, individual cells pass through a laser light. And the laser light tells you the amount of fluorescence of the algae and how many of those algae were stained. We also directly quantified the glycans using mass spectrometry, and this was work done by the Lozgen lab and the McGrath lab. So what we did was we cleaved the glycans off using an enzyme called PNGSF, and then we washed away the algae, just recovered the glycans. And then these glycans were measured using mass spectrometry. So first they were size sorted. So one glycan at a time was analyzed. And so this data tells us the identity of the glycans and how much of the glycans there are. So here's the lectin staining data. We have median fluorescence intensity, which is a proxy or a measure of the lectin staining of the algae. We have ambient temperature, we have heat stress treatments, and then we have CVN and SNA1. And very clearly, we found that high mannose and sialic acid glycans increased with heat stress. So kind of counterintuitive to the original hypothesis, but very interesting nonetheless. And it's interesting because high mannose overabundance has been linked to hindering symbiosis establishment. We know this from comparing different species of algae. Incompatible species have higher amounts of high mannose. We also have done experiments with drug inhibitors manipulating the glycome, and again, an overabundance of high mannose was linked to hindering symbiosis. But the role of sialic acids in symbiosis are largely unknown. We included sialic acids because they have a well-documented role in human biology and human immunity. But otherwise, sialic acids are pretty understudied. So we wanted to include it, wanted to see what was happening. But when we look back at the literature, it's pretty unclear what's happening there for symbiosis. Here's the mass spectrometry data. So there's a lot going on here. These are the individual glycans that we found on the cell surface of the algae. And I'll try to point out some of the big patterns here. First of all, here, these boxes are just blown up versions of these boxes. We just couldn't fit them on the same scale because it was just so small here. So first we have high mannose glycans. And so high mannose glycans were the most abundant of the glycans on the cell surface of the algae. Second most abundant was galactosylated glycans. And then third most, the least abundant, were the sialylated glycan group. And so while we did find differences in the sialylated glycans, their role in biology might be questionable because they're so low in abundance, but we're not entirely sure. What I do want to point out is these galactosylated glycans. So I'm going to blow up here to show you those glycans. So we found some differentially enriched glycans with heat stress. And we found that these two galactosylated glycans were higher in abundance in ambient temperature and lower in heat stress. And so this is interesting because galactosylated glycans have been linked to successful symbioses. So the fact that they have decreased with heat stress is pretty predictive, maybe. And so this evidence comes from native species having higher abundances of galactose compared to incompatible species. And other researchers have also inoculated the algae with the addition of galactose, which has also increased symbiotic uptake. So pretty interesting stuff happening with heat stress. So going back to the question, how does heat stress affect the algal glycome? And we found that the glycome may be altered to be less conducive to symbiosis. So here we have the new model here. We think that with heat stress, the algae are shifting in their algal glycome to become more similar to incompatible species. In this case, they're increasing in abundance of high mannose glycans and decreasing in abundance of the galactosylated glycans. Very importantly, I do want to point out, we weren't able to prove that the differences in the glycans were causing the hindrance in establishment of symbiosis, but the way that they did change is pretty, at least we think there's pretty good evidence that they were involved during the hindrance of symbiosis. Okay, going back to my chapters. So now we're moving on to the maintenance of symbiosis. And I'm going to go a little bit out of order, and I'm going to talk about aptamers first. So chapter 4 is called Identifying Symbiosis-Specific Proteins from Brevulum minutum using aptamer cell selects. And this was the really high-risk project that Virginia was talking about. So before I talk about aptamers, I want to introduce a little bit more about the algal biology. So algae can be ex hospite, which means outside the host, and they can also be in hospite, which means inside the host. And these two environments are very different from one another. So algae in hospite, they're in ambient seawater, and they're independent. They can't rely on their symbiotic hosts. And the algae in hospite, they're in a low pH environment because of the symbiozone environment. The salinity also happens to be low in this environment as well. And the algae have to actively maintain symbiosis and evade immune rejection. So these two environments pose unique biological challenges for the algae. So we believe there's differences in the biology of these algae. And we wanted to know if the cell surface is key for the maintenance of symbiosis. So we've used aptamers to probe the cell surface here and try to figure out what proteins are specific for the symbiotic state. So what are aptamers, right? So aptamers are used for discovery of novel biomarkers on the cell surface of cells. And so aptamers are ligand nucleotides that self-fold and bind specifically to target molecules. So here we have a single-stranded DNA or RNA molecule, so aptamers can be either one. And so they form this structure based on base pairing, so it's a sequence-dependent structure. And then this aptamer can bind to specific targets. And you might not think of DNA as binding to things, but they do so very well, and they do so very specifically as well. And just to clarify, biomarkers are molecules that are indicative of a cell phenotype, such as cancers and diseases. So aptamers have only been used really to study diseases and cancers, discover novel biomarkers for these diseases. But in my case, I wanted to discover symbiosis-specific proteins. So applying a technology from disease biology into symbiosis. Like Virginia mentioned, in the lab, aptamers can be used much like antibodies. They can be fluorescently tagged. They can be used to purify the target molecule so we can discover what's there. And they can also be used for functional studies as well. So they're a very useful molecular tool once you've generated these aptamers. So this brings me to my question for this chapter. Can we use aptamers to discover symbiosis-specific proteins? So to do that, we did this technique called cell selects. So I'll walk you through it. Looks like there's a lot going on here, but it's not too bad. So we first start off with an aptamer library. You can order this from companies. And so we have a naive library. At this point, they just bind to a lot of different things. So first, we incubated it with cultured algae. So this is what a cultured vessel looks like. And so these cultured algae represented algae in the ex hospite free living state. So after the incubation, we discarded the aptamers that bound to the algae along with the algae, and then recovered the aptamers that did not bind to algae in the free living state. Next, we incubated these aptamers to freshly isolated algae. And the way we obtained these, where we ground up a whole anemone, got rid of the host material, just kept the algae. And so these algae represented the symbiotic state, the in-hospite state. So after this incubation, we discarded the aptamers that did not bind and then kept the aptamers that bound to our target cells. So at this point, we have aptamers that should bind to the symbiotic state, but not to the free living state. So these are symbiosis-specific aptamers. And so the aptamers I used were DNA. And because it's DNA, you can PCR amplify and make a bunch of copies of it. And you can repeat this over and over until you get very strongly binding, very specific aptamers. And a considerable amount of time in this chapter was spent on protocol development. There's a lot of troubleshooting in the amplification step. There's a lot of troubleshooting with freshly isolating algae because we don't want any contaminants within this mix. But unfortunately, for the sake of time, I won't be talking about those details. But if you are curious, I'd be happy to share with you those things. So here are the results. We did 6 total rounds of selects, and then candidates were chosen based on sequence abundance and sequence structure. And the sequence abundances we obtained from Illumina high throughput sequencing. So here are those candidate aptamers, and these are the structures that we see here, and they're predictions based on M-fold. So they look like those aptamers we saw in that diagram, which is good, and they are different from one another. Some of these aptamers form pretty robust structures like aptamer 4 there, use most of the sequence to form its structure. Some of them were pretty weak, like aptamer 2, aptamer 9 only used a few nucleotides to form its structure. But structure alone doesn't really tell us what's going on here. So we tested the aptamers and bound them to our target cells to see if they were specific or not. And so to do that, we used qPCR. And so we ordered one aptamer at a time. We folded them correctly into their structures, bound them to our target cells, and then washed away some of the aptamers. Some of them don't bind because of randomness and binding strength. Washed away those aptamers and then recovered the aptamers from those cells and then quantified how much bound using qPCR. So here's that data. So here we have the aptamer concentration on the y-axis, and we have our different target cells here. We have cultured algae and freshly isolated algae, and these are the different aptamer candidates. So you can see here the aptamers did bind to algal cells, which is good, but they were not specific to freshly isolated algae. The binding here is pretty even between the two populations. In fact, aptamer 9, weirdly, bound to cultured algae better than to freshly isolated algae, which was the opposite of what we wanted. And then aptamer 1, you might notice this outlier. If you do remove it, it is significant, but I did repeat this binding experiment with aptamer 1, and it was more like this. It was pretty even. So unfortunately, this didn't work. But we had earlier signs of success. So this is data from round 3 of selects. And so these aptamers were taken from round 3, which is the middle of the selects process. I went through 6 rounds of it. And these aptamers were fluorescently tagged, and it was just a way to check whether they were binding to the cells or not during the entire process. And I did find that they were specific to freshly isolated algae, and I didn't see any binding to cultured algae. And these are representative images, but again, I didn't see any of them binding in the cultured algae. So there was promising results in the middle of it, but eventually it didn't work out. And I was unfortunately unable to repeat this because I found that it took a lot of the material away for selects. So this wasn't repeated. We don't have any more of the round 3 sample. Yeah. So in conclusion, can we use afterwards to discover symbiosis-specific proteins? And the answer is unfortunately not yet, but considerable progress has been made for the development of the protocol so that any future attempts, if people are interested in it, should be possible. And I do have some other ideas for how we can modify this. For example, there's a way to do it without repeating it over and over. You just do it one time and it takes an afternoon. So that would be better to do. Well, I can talk about that, but you can ask that as a question. Okay. So moving on to the next chapter. So chapters 2 and 3, I'll be talking about together because they're complementary to each other. So chapters 2 and 3, we'll be looking at the maintenance of symbiosis and what happens to the algae after they are expelled. So chapter 2 is titled Limitations of Using Cultured Algae to Study Nigerian Algal Symbioses and Suggestions for Future Studies. So this is the perspective piece that I worked on during the pandemic, published in Journal of Ecology, and it was kind of looking at the common practice of using cultured algae for symbiosis studies. It's kind of an introspective thing too, because chapters 1, chapters 4, I did use cultured algae. So going back to this figure about algal biology, again, algae ex hospite, algae in hospite, they have very different life histories. So it's advantageous to compare the biology of the two states to understand symbiosis. So here's a data piece that did just that. This is by Zhang et al. Published in Nature Communications. And so what she did was looked at the transcriptome of the algae in hospite and algae ex hospite. And what she found was nitrate transport and ammonium transporter were the most enriched of these genes in this comparison. And they were both upregulated in hospite. So clearly these transporters are very important for the symbiotic state, but these are indicators that the algae are starved of nutrients. So this data like this, there's plenty of other data that support this as well, leads to the hypothesis that the host controls nutrient availability. So here's what I mean by that. Algae in hospite, or sorry, algae ex hospite, they downregulate their nutrients transporters because there's plenty of nutrients around. They don't need a lot of these transporters to grab what they need. Whereas algae in hospite, they have to upregulate these nutrient transporters because it's hypothesized that the host limits nutrient availability to the symbiont. So they have to upregulate these transporters to grab whatever little there is available to them. But in reality, this comparison uses cultured algae to represent the ex hospite state. And cultured algae are grown in nutrient-rich media because culture are designed to grow the algae as fast as possible, and keep them as happy as possible. Whereas algae in hospite, they're sampled from anemones and corals that are kept in oligotrophic seawater, so a very low nutrient state. So in this comparison, nutrition is a clear confounding variable. So context matters when we're studying these comparisons, especially in the context of symbiosis. So here's that perspective piece, and here's a figure that I made for that perspective, talking about the limitations of using culture. So here we have algae in hospite, they're in oligotrophic seawater, they're in their low pH environment because of the symbiosome, and they have low division rates because it's low nutrients. Algae ex hospite, they can be in the environment, they can also be in culture. In the environment, they're in oligotrophic seawater, they're in ambient pH, and we don't know their division rates because it's understudied. Whereas algae in culture, they're in high nutrient-rich media, they are in ambient pH, but they have really high division rates because they're designed to grow very well. So clearly here, culture does not reflect what is found in nature, right? I should mention here, algae again are released as a homeostatic mechanism. So these are released in a non-stress context. So in this perspective piece, we asked what could be a better representation of algae in the ex hospite state? And we said, what if we sampled algae that were released from the host? Would that control for nutrition as a confounding variable in this comparison? So I did just that. Chapter 3 is titled Algae from Aptasia Egesta are robust representations of symbiotic in the free living state. And then this work is published, or not published, submitted to PeerJ, so hopefully we'll see that soon. But this work is thanks to some co-authors. We have Julia Unsworth, who's here in the crowd here. She's an undergraduate who worked on this and helped gather the gene expression data you're going to see. Val's also a fellow Weiss Lab member who imaged the algae for me. And then Z362 students, these are the invertebrate biology students who also helped collect data, and they appear as a cohort author on this paper as well. Sorry, give me a sec. Okay, so what are egesta? So in Aptasia released algae in discrete pellets called egesta. So here we have the mouth of an anemone. It's a video. And you can see a little piece of egesta coming out really slowly here. I was pretty happy when I saw that. It was pretty cool. And then here we have egesta next to an anemone, just to give you a sense of scale, what the egesta looks like. Here's a close-up of the egesta. You can see the little brown spheres here. Those are individual algae. There's also some protists swimming around here. So again, these are released as a homeostatic mechanism, probably to regulate symbiont density. So again, not a stressed release, and these algae are released daily in the Aptasia. So this brings me to my question: could egesta be a robust experimental model to represent algae in the exhospitase state? Before this study, we really didn't know if the algae were even alive to begin with. So if they weren't alive, then it wouldn't be a good study system. So we looked at a lot of the basic biology of egesta in this paper. We also then asked, what is the gene expression of nutrient transporters in algae from egesta? Could nutrition be controlled for in this comparison? So here's what egesta look like. So this is these pretty images you're about to see are thanks to Val. And these are confocal images. So there's fluorescent images, and these are false colors. So these aren't what they actually look like. But we found that egesta are mostly made up of the algal symbionts. So these magenta little spheres here are the chlorophyll from the algae fluorescing. Each algae also has a little yellow dot, and the yellow dot's a pyrenoid, which is an organelle that glows under certain wavelengths of light, shown in yellow here. But there's other things going on here as well. We found some cyanobacteria. So these little strings here of yellow are cyanobacteria. We found some protists swimming around here, and they glow. I don't remember what color they actually glow, but they are colored cyan here. And so these cyan fluorescence was also seen embedded in the tissue of the egesta, or the egesta itself. And we're not sure what it is, but it might be just the dead form of the protist, but that's just the hypothesis. And there's also some nematocysts. So there were discharged nematocysts on the surface of the egesta. And nematocysts are the stinging cells of cnidarians. We're not sure, maybe this is a way that the host is expelling spent nematocysts, because the anemones can't really recycle that. But it also had made the egesta kind of sticky, which we're not sure if there's any biological significance to that, but just something to note. So we described the basic biology of this and the fluorescence, because in cell biology, it's often advantageous to fluorescently stain the cell. So to document the autofluorescence is useful. And we also did some cell staining to determine the percentage of the algae that were alive. And I don't have images of that, but we found that 98.5% of the algae are alive. So far, egesta is made up of mostly algae, and they're mostly alive. So a good start. Next, we looked at the mitotic index of the algae. And mitotic index is a measure of cell division rates. And it's a very simple way to measure it, because you can tell which algae are dividing because they look like two semicircles smashed together. And so this data was taken by Z362 students. This was done during remote teaching during the pandemic. So what I did was I took a lot of pictures of algae from egesta, took a lot of pictures from anemone tentacles, gave them to the students, and they counted for me in each individual algae, counted all the algae, counted all the dividing cells. And so they collected that data. And altogether, they probably went through 16,000 cells for me. So a great example of crowdsourcing. So here's that data. So this is mitotic index on the y-axis. We have algae from egesta, algae from the host. And these data are paired, which means this egesta came from this host. So in that way, they're paired. And we found significant differences. We found that the mitotic index was higher in egesta than in the host. And this corroborates data, previous data that was found in the literature as well. And we think there's several hypotheses as to why this might be, but we think that it might be that the host is preferentially expelling dividing algae, because if it's a mechanism to regulate symbiont densities, expelling the ones that are dividing is a pretty good way to do that. Next, we looked at symbiosis establishment. So it's pretty similar methods to chapter 1, where you take algae from different sources, added it to an aposymbiotic anemone, and then measure symbiont densities. So here as symbiont densities, we have algae from culture, algae from Egesta, and freshly isolated algae. And we found that algae from Egesta established symbiosis just as well as cultured algae. And we also included freshly isolated algae here because from previous literature, we hypothesized that freshly isolated algae would do better in reestablishing symbiosis. And we thought Egesta would fit somewhere in that middle because they have similar life histories, but there was no statistical significance between any of the 3 treatments. Okay, so going back to the question, could Egesta be a robust experimental model? And it's a resounding yes, and they're very competent symbionts as well. So next we looked at the gene expression of nutrient transporters. And as a reminder, algae and culture, they downregulate their nutrient transporters because there's plenty of nutrients around, whereas algae and hospitae, they upregulate their nutrient transporters because it's hypothesized that the host is limiting the nutrient transfer. So again, this is data collected by Julie Unsworth, so she deserves all the credit for her hard work here. And so we tested gene expression with RT-qPCR. We tested 3 different nutrient transporters, ammonium transporter, nitrate transporter, and purine nucleoside permease. And then we also looked at a nitrogen handler. This was nitrate reductase, which is involved in the conversion of nitrates to ammonia. And this is important because ammonia is the preferred form of nitrogen for the algae. So when they're starved of nutrients, they upregulate this gene to convert as much of the nitrates to ammonia as possible. So again, all of these genes are known to be upregulated in low nutrient conditions. So here we have one gene here. I just want to orient you to this figure before I show you the whole thing. So we have relative expression. So this expression, all this expression is compared to cultured algae. So cultured algae is always at 1. And we found that in algae and Egesta, algae and hospitae, they upregulate ammonium transporter compared to cultured algae. So there's a first sign that the algae are in a low nutrient condition, even in algae and Egesta. And so this pattern held true for all the genes that we tested. For all the nutrient transporters and the nitrate reductase, they were all upregulated in comparison to culture, and there was no difference between algae and Egesta and algae and hospitae. So this causes a shift in thinking of this model, right? So here we have algae x hospitae. This is the old model. Algae x hospitae, they downregulate their nutrient transporters because there's plenty of nutrients around. Whereas algae and hospitae, it was hypothesized that the host is limiting nutrients to the symbionts, so they have to upregulate their transporters. But when you bring Egesta into this picture, even though Egesta are ex hospitae, they have to upregulate their nutrient transfers because there's no nutrients around to begin with. So this comparison here, both the algae in Egesta, algae and hospitae are in oligotrophic seawater. So this comparison can control for nutrition as a confounding variable. And so it might not be that the host is actively limiting nutrients to the algae. It might be that they're just not there, but this will require some further testing to really confirm that hypothesis. So wrapping up here, going back to my chapters. So chapter 1, heat stress of the algal glycone. Heat stress alters the algal glycone to become more like that of incompatible algae. Chapter 4, I looked at aptamers using cell selects, and I found that it's a promising technique for the discovery of symbiosis-specific proteins, and I've made some considerable strides in that protocol. And then chapters 2 and 3, we explored how there's limitations for using culture in studies and found that Egesta is a great alternative to using culture in symbiosis studies. So what is the point of any of this, right? What's the significance of my research? Did I find a groundbreaking discovery that's going to save the corals? We did it, right? No, that's not true. What I did do was contribute to an understanding of the fundamentals of cnidarian algal symbiosis, and small contributions add up together to bubble up to form solutions. At least that's the hope of studying all of this. And my findings also stress the importance of algal cellular physiology on the function of the symbiosis. Oftentimes we forget that the algae are active partners. We just study the corals. But here I found that just changing the algae, just the algae, can have dramatic effects on the symbiosis. Okay, so that brings me to my acknowledgments. So this is my therapist, Dr. Matt Orvell. So thank you, Dr. Matt Orbel, for helping me through the pandemic, helping me throughout the later half of my PhD. I think mental health is really important for the PhD, so I highly recommend getting a therapist. This is my dog, Ami. Thanks for keeping me happy as well. This is my dad, and he's also a scientist, so it's nice to have a family member that I can talk shop with, and he's been very supportive of my PhD process as well. So thank you, Dad. Thank you to my committee. Thank you to Virginia. You've made the PhD experience very fun. And because, of course, PhDs are stressful, but very importantly, you've made it really fun. So thank you, Virginia, for that, because science should be fun, I think. Thank you to my advising committee, Nate, Alan, Jeff, and Andy. You've made my science better. You've made considerable contributions to my work as well. So thank you for your help. Thank you, Nate, especially for helping or allowing me to do the cures, the curated undergraduate research experiments, they're called course-based undergraduate research experiences in the invertebrate biology lab and the introductory biology lab as well. Thank you to the teaching team. Thank you, Lindsay. Thank you, Devin, for really making anatomy a fun teaching experience. I was dreading teaching anatomy when I started this, but anatomy is really cool. So thank you for that. Thanks, Nate, again. Thanks, Carmen. Thanks, Lori, for the introductory biology lab experience, especially during the beginning. It was the first thing I taught. Lori held my hand along the way. So thank you, Lori, for that. And thanks, Nate, and thanks, Carmen, again, for allowing me to go through that cure process in introductory biology. And thank you to all my friends and loved ones. I don't want to go into specifics because I might cry. But thank you for all the support over the years. You've made life a blast and the PhD a blast as well. And thank you for listening.
Okay, thank you, Shumpei. All right, so we have time for some questions from the non-committee members. Dr. Shumpei, maybe on Zoom.
I can see the chat
You
here.
can see the chat.
Yeah.
Okay, so on Zoom or in person.
Yes.
Thanks, Craig.
Hi. Thank you.
Chapter 1, I don't know the algae, species situation, but you say there's a compatible species and incompatible species. Within the compatible ones, what kind of genetic diversity is it? And what I'm thinking is,
Right.
could you screen for higher tolerance and that they don't change their glycans much?
Yeah. So the question was for the Zoom audience, what are about the incompatible species of algae in terms of glycans? You're asking about heat tolerance?
So like even within the compatible one,
Right.
show that heat tolerance increases their the way they can establish.
Yeah.
But is that using one club? Is that using one isolate? Or
Right.
is there genetic diversity within that species?
Oh,
So
okay.
that within that species,
Yeah.
some of them, some isolates, some clonal variation,
Yeah.
may be more tolerant to heat stress, and you can start to select on that.
Yeah. So what we did in my experiment was use a culture, which was just theoretically one genotype. But within the species, there's a lot of different variation, and there are some heat tolerant strains of Breviola minudum, especially those that have been like heat evolved in experiments. We don't know how their glycomes change with heat stress as well, so we don't know. I did some work in Okinawa, which I didn't have time to explore more because of the pandemic, where I took a different species of algae, Geristinium trenchii, which is a heat tolerant species of algae. And they're not affected by heat stress during the onset of symbiosis, but their glycome did change. So that was really interesting. But again, didn't have enough time to explore that. Yeah, and I tested this in coral larvae, so a little bit of a different context, but yeah. Oh, yeah.
So it seems like it's really powerful to be able to use a lot of undergraduates to help you with your work.
Yeah.
Yeah, because they can collect a lot of data. How What are the ways in which you had to manage that to make sure you get reliable?
Yeah,
data
yeah,
from the
yeah.
undergrad?
Yeah.
And do you think you'll keep investigating that path of cures for
Yeah.
undergrad research?
So the question was, how do I manage cures in a group of undergrads to get quality data? And to do that, so what they did with, they counted the cells with ImageJ. And so there's like pointers or like flags that they put on the images. So I looked back at those manually to check, and they actually did a not so great job. But it did make it a lot easier because they clicked each cell at least. So I went back and corrected their counts. And so I'm confident about the quality of the data because I did verify it. But also I had 2 students look at the same image. So in that way, I was also able to try to get quality data from that as well. And the other question was, do I plan to incorporate cures through my life, career? And yeah, definitely. I think cures are pretty powerful. Teaching is fun too. And teaching with cures can really inspire students to get into research and understand original research as well. So yeah, I definitely plan to do that. And I have one planned for my postdoc. I'll be coming back here actually for a few months or maybe a month. But anyway, should I take a
Yeah.
Zoom question? This Olivia asks, do you have to stimulate aptasia to expel the egesta, or how do you obtain it? The answer is, so I looked at, I didn't show the data, but I found that algae or the egesta were expelled at night. So in that way, I can time when to collect the very fresh algae. But I didn't do any stimulation to release the algae. So what I did was just came in the morning and collected them, and they were pretty fresh. I washed the containers before. Here's another question from Jack. Do you think you could simulate the in-hospite environment to prep algae for inoculation, or for studying the in-hospite environment ex hospite? Interesting. Yeah, you could maybe not the in-hospite environment. Oh, like maybe if you lower the pH, that might be interesting. Yeah, but I think studies should, if they are going to use culture, maybe lower the nutrients for a week to control for nutrition as a confounding variable. I think that would be a pretty easy way to control for that. But yeah, those are things to think about for sure.
We probably have time for one.
Okay.
We actually don't have. Do we have
Okay.
one more?
One more. Yeah.
So is your ultimate goal, this is a question coming very ignorant, but is your ultimate goal to be able to regenerate quality, no matter the temperature, but in the rate to which your environment or acid environment?
That's a good question. What is my ultimate goal? I think making corals more durable to heat stress might be a good thing to do. There is a problem with scalability, but at local scales, it's very possible. You can try to get corals, identify corals that are very heat tolerant, try to bring those, maybe plant some of them, maybe bring them in somehow or another. There's a lot of different ideas. But yeah, for now, I'm just trying to understand the basics of the symbiosis. Yeah.
Okay, great. Thank you so much, Shumpei, and the committee will follow us before they call. Thank you.
Thank you.